Okay, good afternoon to everybody. I hope you can hear me. And just give us out to confirm that you can hear me.
Yes, sir.
Okay.
Yes, sir.
Yes, sir.
I'm Dr. Amit Sarkar. I'm Head Department of Biotechnology, and I will be taking this course of the basics of immunology. First of all, I don't know what is going on here. I was initially there was 30 students, and now I can see only 8. Some presenters is 7 and attendees 35. Anyway, so I know that it's a very weird situation for both you and me. I think you didn't get the chance to go to the university since you joined a university last year. And that's why you do not know me and I do not know you. And you also do not know your classmates. But there is no point in complaining. We have to continue in this way as long as the situation is not restored to normalcy. Here in this class, there will be 54 students total. 29 students from agriculture, the maximum. Then 16 students from physiotherapy, 4 each from pharmacy and BMLT, and 1 for fisheries science. I will check the names little later to see that everybody's name got included in this meeting. Today, before I begin, I would like to address something. First of all, I know that this is your first GE class. You will get a chance to opt for more Gs in your subsequent semesters, up to 4 or 6 semesters, I suppose. So what is the G? Maybe somebody has briefed you, or I do not know, I'm not sure. So G or general elective subject that comes under the UGC recommendation of CBCS, or choice-based credit system. That means you can make a choice. So that means you have chosen this subject. You could have chosen something else. So the aims and objectives of offering a G subject is that a student should get a chance to study something which is not directly related to their subject. Here, all of you are more or less coming from life science related subjects, so I can say that a bit related. But you can imagine a situation when a computer students or mechanical engineering students may also opt for the same subject. There is no relation. So there's a chance that this knowledge might not help in your career development as per se. But developing the career is not the only thing. We study things to gain knowledge, which could be applicable in our life for the betterment of society. So that's why the G subject, this concept has came into effect in last couple of years. So is there a difference from core subject and G? Of course, there's a difference. Core subject cannot be compared with the G subject. But from the teaching point of view, there is not much difference. Only one difference is that I will not go maybe as much as in detail, which I'm supposed to go for a biotechnology student. That is quite understandable. Otherwise, there is no difference. But from the teaching point, from my class, my point and my objective is that being a G subject, you must enjoy the class. Whatever I am going to discuss, whatever I'm going to learn, that experience will be enjoyable. That giving a lecture for hours and hours and making yourself bored, that is not going to be my objective. I will try my best not to do that. But as I said, just like in a core class, it will be also 1.5 hours is allotted. Maybe I will go for 1 hour, or sometime I will go for 1.5 hours. There will be exams, of course. Internal, there will be 2 internal exams, at least. There will be at least 1 or 2 assignments, home assignments. There could be some quiz sometime. And you know that marks are allotted for each and every section of this type of things, apart from the end semester exams. And you also know that attendance also provides some marks, 5 marks for attendance also there. Last year, 8 students or 9 students are there only in my class. Unfortunately, 3 of them failed. So I'm not saying that to scare you. They failed because they didn't attend the classes at all. Whatever reasons, that's why they failed. Otherwise, there should not be any reason that it will fail. So I will suggest not to go into that footpath. You must attend the classes regularly as much as possible. Missing 1 or 2 classes here and there is okay. I'm absolutely fine with that. So that this type of not getting good marks or failing as question should not arise. It will be totally very annoying thing for you when you'll be asked to appear for a supplementary exam for G paper. Okay. I will take this class on Wednesday to let you know, but there is a slight chance of time change for 1 month period. There's a chance that I will go for a training. If I go, then the time will be changed for 1 month, 4 classes. As per the university rule goes, I should not provide you any notes. I will not do that, but I will share the PPT files I am going to present. That will help you. Besides that, I'll also provide you any ebooks. If you like to, you can go through that ebooks. Yes, Aditya Mukherjee, I will take questions. Let me finish my thing, then I will take your questions. I will take all the questions, Ovik and Aditya. Let me finish my talk, then I will ask for your questions. So I will be providing my phone number and my email. I already provided in the team weeks before. So that will be one mode of communication. Another mode of communication that I think you'll be more comfortable with, that's a WhatsApp group. So we'll form a WhatsApp group. I've already formed it, but as your number is not in my contact list, I'll give you an invitation link here, and you can join using that invitation link easily. So we'll try to make our communication very effective through that WhatsApp group. Whatever questions or any queries might arise, you can communicate, and I will also do the same. During the class, you can ask questions at any point of time, but I will go with a full screen mode thing, so I will be able to see who is asking the question, but that's okay. You can tell your name and then you can ask the questions. That's not a problem. And regarding the attendance, I'll provide you a Microsoft Teams form at the beginning or the middle of the class. You will given 15 minutes of time for that, where you have to put your name, date, and time, and you have to submit. Later, I will convert that form into our ERP system. So that will be my record. If somehow any technical problem arises, you can communicate with me that this is the reason I couldn't put my attendance. That will be considered. No issue about that. So overall, as I said, our target would be to make it a very enjoyable experience. Okay, so now anybody can ask any questions if you have before I start other things. You can ask me. Ovik, you're asking something, and somebody else also. Please shout.
স্যার আপনি নোট প্রোভাইডের কথা বললেন, ইউনিভার্সিটির রুলস তো আবার চেঞ্জ হয়েছে, তো নোট প্রোভাইড করা হবে সেটা শুনলাম। মানে আমাদের প্রোভাইড করা হচ্ছে,
ওরকম কোনো রুল হয়নি, হবেও না। তবে যে তোমাকে বলেছে সে ভুল বলছে। তবে আমি তোমাকে পাওয়ারপয়েন্ট স্লাইডস দেবো, সেটা তোমাকে হেল্প করবে, কোনো অসুবিধা হবে না। দ্যাট উইল বি অ্যাজ ইকুইভ্যালেন্ট টু দ্য নোট। ওকে, বাট ইট উইল বি অলসো অ্যাডভাইজেবল দ্যাট ইউ study the books. That will be helpful for you. And let me see. So here I'm going to send you the invitation link for the WhatsApp. Now try to use the invitation link and try to join into the group. Let's see whether you can do that or not. Yes, I'm getting response. 3 people already joined, 4, so it's working. Fine. So you can share with your friends who are not present here today, maybe, if they want to join in this group. So as I do not know your names, I cannot match your name with the contact number. So if you have a question, just tell me your name and the stream. Okay. And as I said, I'm going to read out the names who supposedly should be here in this class. So this is not an attendance call, so you need not to respond. Just check that your name is there or not. Okay. So I have one student from Fisheries Science. The name is Priyamedha Shaha. 4 students from Pharmacy, Shohom Dash, Kaushik Giri, Saurabh Maiti, and Padmanabh Seth. 29 students from Agriculture. Names are Ananya Chakraborty, Onik Sen, Abhishek Lashkar, Ayon De, Mouli Chakraborty, Nishak Roy, Aditi Kirtania, Pushpita Sarkar, Shreyoshi Das, Sneha Sutrodhar, Sudipto Das, Tania Khatun, Arnab Adhikari, Avik Jana, Nilabhro Metia, Monalisha Ghosh, Shayok Karmokar, Aheli Singho, Aditya Mukherjee, Aritro Mondol, Neha Jana, Saurabh Biswas, Tanvir Islam, Konkona Sarkar, Saurabh De, Eshika Bera, Papia Boisho, Anisha Maiti, and Shayok Dotto. 16 students from Physiotherapy, Priyangshu Das, Shaumodev Sautto, Shushovan Saha, Durbadal Adhikari, Shoujanno Samanta, Jyotirmoy Goswami, Shaumo Banerjee, Murali Samanta, Supriya Parveen, Billodal Ghosh, Shafia Khatun, Mohima Khatun, Kausar Kalim, Arnab Basu, Neha Mandal, and Shayan Modak. 4 students from BMLT, Salman SK, Muhammad Ali Khan, Saurabh Bera, and Rakibul Haq. Now tell me if I haven't mentioned anybody's name. Is there anybody whose name has not been mentioned? I suppose not. I have 46 students here, so 8 students are not attending this class. That's okay. So, do you have any more questions, general questions? I think everybody could use that WhatsApp link. I can see a lot of people have joined. That's great. So far, 36, so 35 of you have joined out of these 46. Fine. Now I am sending the attendance form link to your chat box. Just give me time. So the attendance form will not accept any response after 15 minutes of time. So that is more than enough. So here is the link. Confirm me if you can open the form. Can you open it?
Yes, sir.
Yes, sir.
Okay.
Opening.
So provide your attendance and we will start the teaching portion in 5 minutes time.
Sir, sir,
Yes, কে বলছো?
স্যার আমি নিশক বলছিলাম।
বলো।
স্যার টাইমটা কি এখনই মেনশন করব?
হ্যাঁ, যে টাইমে তুমি দিচ্ছ সে টাইমটা মেনশন করো।
আচ্ছা, ওকে স্যার, থ্যাংক ইউ।
Can you see the PowerPoint? Please confirm me.
Yes, sir.
Okay.
Yes, sir. Yes, sir.
So
Yes, sir.
I'm going into the full screen mode. And just one thing I forgot to mention, that the instruction medium will be totally English. Our question answer session also will be preferably in English. But of course, I will go slow and I will use very simple English. That should not be a problem for you. Okay, let's see. So today is just an introductory class. We will not go into much
Hello, sir.
detail. Yes.
স্যার
বলো।
আপনি একটু বাংলাতেও প্লিজ ট্রান্সলেট করে দেবেন।
No, Bangla translate করা যাবে না. You have to learn to understand English. If you have any problem, you can ask. I will repeat that. That is okay. But no vernacular language will be used in this class. You have to learn to understand English lecture. But of course, you have every right to ask me that if I do not understand this point, can you please repeat? I will try to do that. Okay. So basics of immunology, that is the name of this course. Very good. So I am pretty sure that in this COVID pandemic situation in the last 1.5 years almost, you have heard about, this is the most used term you have heard: immunity, immunology, immune response, antibody, antigen, vaccine, etc. So we're not going to talk about the COVID unless it's important in some example or so. So we're talking about in general immune system, of course. So immune system is considered as a remarkably versatile defense system. Versatile, that means it has several different parts or components. Defense system against what? What the defending against? Defending against invading pathogenic microorganism. So huge number of microorganisms are there in our biosphere, and this defense system is actually protecting us against those microorganisms. But we are only concerned about those which are pathogenic one. Pathogenic. So if they're not pathogenic, they're not causing any disease to us, so we are not concerned, and our immune system do not need to provide any defense. Say, for example, we know that we have tons of bacteria in our stomach or intestine. Those are beneficial bacteria, good bacteria. So those are not pathogenic. So our immune system is not attacking those bacteria. So remarkably versatile defense system that has evolved. That means the immune system was not in place million years ago. Gradually it's evolved, and it's still evolving, and it will evolve in future too. It's a continuous process. Now, as I was saying, that microorganism theoretically, actually, the numbers are limitless. We, first of all, who are the microorganisms? We will consider as virus, bacteria, parasite, fungi, and also helminthes. Helminthes is not microorganism. Helminthes means kecho, creamy, etc. So those are not microorganisms, but still our immune system gave protection to them. The thing is that how many number of bacteria and virus we have in our biosphere, that is not known to us. So rather we should say that limitless. Every year people are discovering thousands and thousands of new types of bacteria, new types of virus, new types of parasite or fungi. So you can imagine that in an enemy front, huge number of any microorganism or pathogenic microorganism is there. And we have one system. So only one system is fighting against these limitless enemies in terms of microorganisms. You must have noticed another thing that is also says that immune system provides protection against cancer. Yes, that is true. But that is, we're not going to touch that front. If sometime I get some time, I can casually mention something about those things. But mostly I will focus about this invading pathogenic microorganism. Good. Now, immune system. There are several different organs are there in our body which are considered as a part of immune system. Different types of lymph nodes, such as tonsil. The tonsil position is totally wrong over here. Tonsils would be something around like here. Lymphatic other vessels, lymph nodes found in the underarm, in the groin area, also the appendix, the bone marrow, thymus gland, spleen, all these comprises immune system. Now this immune system, they produce several different kinds of immune cells. So here the list of immune cells you can see: monocyte, mast cell, eosinophil, natural killer cell, dendritic cell, basophil, neutrophil, lymphocyte, macrophage, platelets. So one thing you can notice over here that all these things are actually blood cells or derivatives of blood cells. But which one is not present here among the blood cell? Red blood cell. Red blood cell has no role to play in our immunity. Only the white blood cell or different derivatives of white blood cell are considered as cells of immune system. So immune system can generate enormous variety of this type of cells, but not only cells, they also produce different types of chemicals like this: complement, chemokines, cytokines, and of course antibody. I'm pretty sure that you've heard about antibody. All of you must have studied in the higher secondary class as well, but maybe you haven't heard about these chemokines, complement, cytokines, and other type of chemicals called interleukins, etc. Okay, so immune system is generating two types of things: cells. These are the cells, not one single cell, several different cells with different job, different role, and also a variety of chemicals like complement, antibody, cytokine, chemokine, etc. Now, what they're going to do, these chemicals and these cells, what are their role? Their role, we understand, is to eliminate those foreign invaders that I just mentioned. Limitless number of foreign invaders who can enter into our body through different routes. Some can enter through our food, which is called foodborne pathogen. Some can enter through our water, that's a waterborne pathogen, or from air, like the COVID, something like that, or through the droplets. So these are the different ways they can enter. They can enter sometime when you get a cut in our skin or something like that due to accident. So they are entering, and these chemicals or cells are eliminating them. Eliminating, there I will see gradually in a later class how they eliminate, whether they kill them always or not, etc. But there is another point, another word is also mentioned over here, recognizing and eliminating. So first, you have to recognize one, that this is indeed a foreign invader. I will come and let's discuss so that point. So these cells and molecules that I have mentioned, they do not work separately. They work in a cooperative manner. They help each other. They communicate with each other. Communicate means, of course, cells cannot talk in English or Bengali or Hindi or anything like that, but they can communicate with each other through chemicals, through chemical signaling. So these cells and molecules, they act together. And that's why they form a very complex network. So this is just a representative figure. The complexity is much, much more bigger. And that complexity only can be comparable with the nervous system. We know that our brain is a very complex system, organ. We do not know all the things, all the features of a brain. We're still in our infancy. We're trying to understand our brain. So that's a very complex, and also the nervous system goes to the spine. So immune system complexity can be compared with that nervous system. Similarly here also, just a second. প্রশ্ন আমি একটা ক্লাসে আছি, সন্ধ্যাবেলার দিকে কথা বলবো, ঠিক আছে? Yes. So similarly, immune system is also comparatively a new subject. If you compare it to the physiology, if you compare to the biochemistry, immunology developed much later, and it's still developing. We know quite a bit, a good amount of information we have about immunology or immune system, but still there are lots of gray areas. So cells and molecules forming a complex dynamic network. Now, as I said, recognition. So we can say that generally an immune response means when an invading organism is entering into your body, how your immune system is addressing the problem. Some problem is there, some bacteria entered in your system through food, maybe you have somehow accidentally eaten some spoiled food and some salmonella or staphylococcus entered in your body, and how your immune system is addressing the problem. So that is called immune response. But that response generally could be divided in functional point of view in two parts: recognition and response. Recognition and response. Why recognition is coming? Because as I say, tons of different types of bacteria are always entering into our body, but all of them are not pathogenic in nature. Some of them are good, beneficial. Some of them may be not neither beneficial nor harmful. And now if you are consuming a bowl of curd, doi, so you know that millions of lactobacillus bacteria are there in the doi or curd. So those are good bacteria. Okay. So now you see, and maybe in that curd, some bad bacteria are also there. They also entered in your system. So now you see it's a problem for the immune system to recognize which one is a good one, which one is a bad one. So maybe you can think that somebody in your house started shouting a thief, 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 chor, chor, and you found that yes, somebody is actually moving in your room, in your house, totally wrapped in a black cloth or something like that. And everybody started hurling abusive language to that person. Maybe somebody is hurting that person. And later you found that that is actually your maternal uncle. He was just trying to create do something funny, or just cutting a prank or something like that. And you unnecessarily, unwantedly, and unintentionally actually did a bad thing to your maternal uncle. So that's why recognition is a very important thing. You have to, the immune system have to recognize which is bad, which is not bad. So you see, immune cell never actually attacks our own body cells. They only attacks or take actions against the foreign invader cells. Foreign cells, and also it should have foreign pathogenic cells. So not only that thing, that is only not recognizing between bad and good bacteria or virus, it's also discriminating between our own cells. So we have our organ with a different type of cell, nerve cell, spleen cell, liver cell. Principally, they all look like the same, like a bacteria or something like that. Maybe a bigger in the size. So immune system should not attack our own cells, because that will be a problem. People will die if immune cells start attacking our own cells and kill them or destroy them. So that is an unique feature of immune system. The system is able to discriminate between foreign molecule and also the body's own cell. But what is how they do that? They do that based on what kind of protein they have. And the difference is very subtle. You know that proteins are made of amino acids. 20 different amino acids are there, which comprises all the proteins. So only very slight difference are there between a bacterial cell and our own liver cell, and also a lactobacillus, that's a good bacteria. But only that small difference is good enough for our immune system to recognize that whether it's a foreign invader pathogenic microorganism or not. Okay, so that's a remarkable feature. Otherwise, our existence would have been difficult if our immune system starts attacking our own cell. It unfortunately happens to some people, which gives rise to a disease called autoimmune disease. That is not a single disease. There are lots of different types of disease out there which comes under the purview of autoimmune disease. Say, for example, myasthenia gravis is there, psoriasis is there, multiple sclerosis is there, etc. So what they're doing, they're recognizing between our own cells and foreign cells, etc. But similarly, as I said, they can distinguish one foreign pathogen from another. That means immune system. So as I said, limitless type of microorganisms are there. So say, just think about bacteria. There are also millions different types of bacteria. But immune system can understand that, well, this is a salmonella, this is an E. coli, this is an staphylococcus. They can differentiate between them. Okay? That have an advantage and disadvantages too, I will discuss later. Not only that, if you study the microbiology, you will see that one E. coli strain, E. coli bacterium, has more than 200 different types of variants or strains. Now, you must be very much familiar with the variant term, as you have seen that the COVID has different variant, alpha, beta, gamma, delta, lambda, kappa, etc. Similarly, bacteria has also different variants. Immune system is so specific about the recognition property that they can almost recognize each and every one of them separately. So all 200 types of E. coli are separate entity for the immune system. all 170, say for example, different strains of Salmonella are different entity for them. They can recognize them separately, and separately. So that's why it's called a remarkable feature of immune system. So we're discussing that our immune response has two functional activity. First is recognition that you have started to understand. Of course, we'll discuss later also. Second thing is that response. So once a foreign organism has been recognized, so once if you think about yourself as an immune system, so now you are confident, you have confirmed that yes, this is indeed a foreign organism. I have to eliminate, I have to neutralize this particular microorganism. Then the immune system recruits those cells that just I have mentioned before here, these cells and also these chemicals. Okay. So once the recognition part is done, then comes the response. Recruits a variety of cells and molecules to mount an appropriate response. So every word has some meaning. Appropriate, that means response is not a generalized thing. So if this is a chichke chore, maybe one tight slap will be enough for that chore. He will flee, go away from your house. Or maybe if I shout very strongly, then he will go away from your house and you will never come back or something like that. But if it is somebody else who has some weapon, then maybe you have to be very careful about that. Okay, maybe you have to call the police to deal with the fact. So all the responses are not the same. Whatever response is for E. coli may not be same for Salmonella, may not be same for a virus, it will be different. So once a foreign organism has been recognized, the immune system recruits a variety of cells and molecules to mount an appropriate response, which we call effector response. So actually the response, the full term of the response is effector response, to eliminate or neutralize the foreign organism. As I said, whether the organism will be killed, that means eliminated, or neutralized. Neutralized means the bacterium will or virus will not be able to replicate again. So say thousands of bacteria entered in your body. Maybe you know that thousands of bacteria who entered in your body, most of the time they cannot do much harm to your health. The problem is that within a very short period of time, they will grow in a geometric progression and there will be huge number of bacteria will be produced. And that might cause a serious problem to your health. But if somehow our immune system can stop that thing, stop replicating, then the 1,000 bacterium will not be able to replicate. So 1,000 bacterium will remain as 1,000 in 5 minutes time or half an hour time. And generally, the half-life of the bacteria is very short. So they will die within a short period of time and you will not feel anything. So such type of things are always happening in the background. You are surviving, you are doing this and that. Maybe you are maintaining hygiene or not maintaining hygiene all the time. So by knowingly or unknowingly, most of the time unknowingly, you are allowing huge amount of microorganisms to enter into your body. But in your background, in your body, there is a robust immune system. A defender is there. If you're a football fan, you can think about a central defender is there who are trying his best to protect you. And in 99.99% time, the immune system is actually successful. So that's why you are not feeling anything. You are not getting sick. So if you imagine a situation when immune system is not there, somehow it's malfunctioning, it's not working due to some genetic problem with that. Unfortunately, some people in this world also suffer from some disease. So it's almost impossible for a person to survive. Every day, every passing moment, you will get infected and there will be some problem, some issues, and people will not survive after a certain age or something like that. Okay, so recognition and response. So in this way, we found that our immune system is converting the initial recognition event, converting it to the response. At first recognizing it and then responding. Okay, so conversion. So you can think about some mechanical energy converted to the electrical energy, something like that. Okay, or heat energy in thermal plant. When we burn coal, we get electrical energy. So it's conversion of heat energy to electrical energy, something like that. Conversion of the recognition event into the response event. And most importantly, each uniquely suited for eliminating a particular type of pathogen. So now I hope you understand the complexity, the amount of the extent of this system. On the one hand, we're saying that we do not even have any idea that how many numbers of microorganisms are there in our biosphere, in our earth. On the other hand, we're saying that our immune system has a response system uniquely suited for eliminating each and every particular type of pathogen. That clearly proves the complexity of the system. Okay. So one last thing at this section, we will discuss later in much more elaborate way. So you see, it's not that you only one time you are getting exposed to a particular microorganism. You have, maybe you are taking tea from a roadside stall or biscuit from a roadside stall or something like that. Maybe the tea vendor is not maintaining a good hygiene or something like that. Or you didn't wash your hand properly. So you have consumed some bad bacteria. Okay. And you suffered a little bit of diarrheal disease. That's okay. We all suffer from such things. We do not care that about much. We know that it will go away in 1 or 2 days. Then again, after a couple of days or 1 week, you went to the same tea stall and you didn't care and you got exposed to or you consumed the same kind of bacteria again. So that's called a later exposure or second exposure. This way, second, third, fourth exposure can go on because more or less in our country or in our state in West Bengal, we are more or less habituated in those type of practice. We take tea from outside stall, we take puchka or something like that from the roadside stall, we do not mind. So the later exposure to the same foreign organism induces a memory response. That means the immune system will be able to remember the microorganism who came last time. So what will be the benefit and what is the meaning? So you see, I can give you another example. So say you are throwing a party in your house and have invited only 20, 25 of your very close friends. It's a very close party. And everybody in the party, I forgot about that. So now you found that one of the person, he is not familiar to you. So when you challenge that person, you found that person has crashed the party. He entered into the party without any invitation. So maybe you showed him the door, you asked him to leave the party, obviously. But you remember the face. First time, you were a bit confused because you were a bit skeptical. That should I challenge that person? Is he my friend? Somehow I'm getting forgetting his face. So if I ask him or challenge him, he might feel insulted. So you're a bit hesitant. When you become a little bit confident, then you ask that person to show his identity or ask him to tell his name, etc. And you found that yes, he's indeed an invader. And now you can imagine that after 3 months of time, you again hosting another party in your house with the same group of friends. And again, that person cast the party and entered in your house. But this time what will happen? You will immediately recognize the person because you have a memory. You now memorized his face. It will not be, it will take a few seconds time. I know that person. This is the same person who invaded my house 3 months back. And this time maybe you will be very angry. Because why this person is constantly again and again invading a party and coming into a party without invitation. So your action could be a little bit more harsh. Maybe you can call a police or something like that. Here also similarly, if repeatedly an organism comes into the picture, then it helps our immune system to develop a memory. An advantage is that when later time it will come, the second time, a third time, our immune response, first of all, will be very quick. There is no time required for analyzing its chemical structure or anything. It can readily recognize that. We will see later how we do that. You have to remember that we should not learn immunology related thing from paper, newspaper. So there is actually no memory. That means they do not have any brain or anything like that, that they can memorize things, they can recite poems from their memory, something like, not something like that. But we use the term memory response, of course. So first advantage is that response will be very quick. And second is called heightened. Heightened, we'll see later a graph or something like that. So as I said, your response to the invader was very harsh, something like that. Or in scientific word, you can say that more antibody will be produced in a short period of time. That will help you to eliminate the pathogen and prevent the disease. Maybe you understand that is more or less the basic understanding about the vaccine development also. Okay, so that is about recognize and response. Just give me a shout before I go into the different section. How is it going? Say something.
Good, sir.
Good. Okay. So these are not just like introductory words of thing. We'll see some historical perspectives of immunology or development of this subject. As such, to be frank, history doesn't have much meaning to scientific discovery. But it's also very good to know how the subject developed, what are the people who mainly contributed. It gives us a feeling that we know the legacy of the subject, how they developed, etc. And in case of immunology, historical perspectives are quite interesting. So what happened first? This particular terminology, immunology, or immunity, it came from a Latin word, then immunios. That means exempt. I hope you know understand the meaning of exempt. You are exempted from appearing for the exam. I can say you are such a good student, you do not have to appear for the exam. I can give you 10 out of 10 if somebody does that. Of course, that is not a practice. So you are exempted. So something like that, exempt. That is the word, English word immunity developed, meaning the state of protection from infectious disease. So in terms of COVID perspective, you can say, or at least you can hope something like that. Maybe that is not entirely true, that who are vaccinated are exempted from the infection. Okay, that is not true entirely, but that is our best hope. So how is the term observed? Actually, for a very long time, people observed that for certain infectious disease, I hope you know the meaning of infectious disease. That the Bengali is Sankramak. Okay, that can be contaminated from one person to another. So individuals who had recovered from certain infectious disease were thereafter protected from the disease. That means once you have suffered from a particular disease and you also recovered, you survived, you are not going to get the same disease again. That was an observation was going on much earlier in the BC time. People don't have any idea why, how, or why this is happening, but that was a general observation. Recovered from certain infectious disease, thereafter protected from the disease. That same notion still goes on in our society today. If we see somebody got infected with or suffered from measles, mumps, or chicken pox, etc., we generally think that that person is got protected. He or she is not going to get the same disease again, at least in a short period of time. There is a historian known as Thucydides in 430 BC. You can imagine this is almost 2,500 years back. He described in a book that plague disease, plague is a very devastating disease, happened in Athens, Greece at that time. And the problem is that so many people got sick at the same time that it was difficult that who will nurse others. And people are very scared. Something like a COVID. Should I go for help? What will happen if I get contaminated or something like that? So it was found that who had already recovered from the plague, who suffered from the disease and then recovered somehow, they are actually taking the nursing job. Because of that same notion, as I mentioned here, who had recovered from certain infectious disease, thereafter protected from that disease. You actually do not to go for that far. Maybe you've heard about a disease called Ebola. It's also a virus-mediated disease. Fortunately, we haven't seen any case of Ebola in India. Ebola mostly affected Middle African country known as Congo. So Ebola still do not have much medicine or vaccine, etc. And people may die within a very quick period of time due to this virus. Now, in the Congo is a very poor country with a very poor health infrastructure, etc. There was a time in 2016, 17, I suppose, no, not 17, 12, 13 or something like that. So many people are getting affected in one village or one city, the same disease. Who will going to be nursing them? Who will be the doctor? Everybody was scared. At that time also, those people came forward, or they've been asked or encouraged to come, who already had Ebola and survived somehow. So now you can consider yourself that I'm more or less protected. But that is a modern era. We know the reason now. But at that time, the reason was not known that why this is happening. So early societies, they recognized this phenomenon, but nothing happened. Almost 2,000 years. Nobody could shed any light on that fact, that why this is true, why somebody is getting protected once they got the disease and survived. So 2,000 years almost passed in a latency before the concept was successfully converted into a scientific concept. And then, of course, it reached to the doctor's table or medically effective practice. That means we know for a long time this phenomenon, but we had no idea why this is happening. No scientific work has been done onto this particular observation. Here first, you must have learned about this person as in a higher secondary class. Edward Jenner, 1798, was a doctor. Edward Jenner observed the same thing as I just mentioned, but about a particular disease. That's called smallpox. You must have idea about the disease. The smallpox used to be a devastating disease once upon a time. Not very long back, 100, 120 years or so. Village after village have been wiped out due to that particular disease, smallpox. there was no medicine available for the smallpox. But later, vaccine is developed due to the smallpox, and that is one of the remarkable feat for the human civilization, that they have wiped out the smallpox from this earth. There is no incidence of smallpox happened after 1970s, no natural infection. Smallpox only exists in some high security laboratories for academic purpose in Russia, in US, in such countries. So you see, in the 1798, smallpox was a very dangerous disease. People are dying in a huge number. Then another disease also was going on in parallel, which is known as cowpox. That means this pox usually used to affect the cattle, cow, buffaloes, etc. Cows used to suffer, but it was found that the people who are dealing with the cows, that means the maids, milkman or milkmaids, they are coming very close contact to the animals, and they are also getting the cowpox. Symptoms of cowpox a bit similar to the smallpox, a rash, a little bit of fever, and that's all. Cowpox was not at all dangerous for the human being. There is no comparison to the smallpox. So milkmaids, the women milkmen, actually, they used to take care of the animals. They're getting the cowpox, and that gets recovered in 5 or 7 days without any problem. But a good quality, an extraordinary quality of a scientist is that a keen observation. But Jenner observed that those milkmaids who had the cowpox disease, when they're also getting the smallpox, they're not suffering that much. They're not getting killed by the smallpox. I hope you understand what I am saying. So this is a milkmaid getting the cowpox, recovering from the cowpox, and then later, when the dangerous actual smallpox is coming into they're getting infected with smallpox, they're again suffering from the disease of smallpox, bit high fever or something like that, but mostly they're not dying. Okay, they are not suffering that much. So that was the from the smallpox. Okay, but why? Of course, Jenner has no idea about that, still relying on simple observation. Then Dr. Jenner did a very abnormal type of experiment that is not ethically should have been done, but he did. Maybe just because he was a doctor, he took that risk or something like that. What he did, he at first inoculated an 8-year-old boy with a fluid from cowpox pustule. Pustule, you can think about foshka, what happens. So in case of cowpox, you will see this type of pustules in the skin. So Jenner extracted the liquid from the pustules. That means some of the cowpox virus will be there, and then injected those liquid into an 8-year-old boy. Fine. That 8-year-old boy suffered from mild disease, and this is a cowpox, not going to be dangerous. Then, most importantly and interestingly, the latter intentionally infected the child with actual smallpox virus. Now you see the amount of risk he took at that time, because there was no medicine for the smallpox available, but you are intentionally infecting somebody artificially with the smallpox. And as a doctor, you know that there is a high possibility that the child will die. So I still personally do not understand how he took that risk. And of course, as I said, ethically, that is not permissible because 8-year-old boy is a minor. He cannot give his consent for this type of experiment. But anyway, that happened in the medical history in 1798. At that time, this type of ethical thing was not much in place. And maybe Edward Jenner was coming from a very high hierarchy of the higher rank of the society. As a doctor, he could have been done anything he wished to do. But he did that. I only wonder that how he spent his night when he knew, maybe only he knew that what he did, he intentionally infected the child with a smallpox virus. And what is his defense? His only observation that milkmaids who had contracted the mild disease of the cowpox were subsequently immune to smallpox. So just based on that observation, he took that challenge and did that experiment. But fortunately, as predicted by Dr. Jenner, the child did not develop much of the smallpox symptom. The boy survived. So in a book, all the names of everything has been mentioned. So Sarah Nelmes was a milkmaid infected with cowpox, and the boy's name was James Phipps, inoculated with cowpox pus, a pustule fluid from the Nelmes, and then Phipps falls ill with a mild case of cowpox. Then scabs are collected from a smallpox patient, and Phipps is inoculated with the scabs of the smallpox, and Phipps is unaffected, and protection is complete. So this experiment is of course a great milestone in the immunology subject. For the first time, somebody experimentally tried to prove what was observed for thousands of years. But one thing you have to keep in mind that Edward Jenner also had no idea what is going on, why the boy got protected, no idea. Anyway, but still, Edward Jenner's experiment is considered as a milestone. Then came the person, you all know of the name. He did a lot of different types of scientific work in different fields. Louis Pasteur, a French scientist, in 1879. So there's a big difference in the timeline. 1798 and then 1879, 100 years almost. Louis Pasteur and his contribution in immunology is kind of accidental. Louis Pasteur basically was a microbiologist who used to work on microorganisms, but not much about the immunological part. At that time, one particular disease called fowl cholera. Fowl means, you know, the cocks and hens and pigeons, etc. are called fowl. So birds were suffering from cholera disease. They're suffering from diarrhea and then getting died. So nobody was aware of the fact that which particular organism is responsible for this fowl cholera. So Louis Pasteur, for the first time, cultured the excreta of those birds, stool of those birds, and isolated the bacteria and identified that Pseudomonas maltosida is the name of a bacteria that is responsible for this particular disease. He also did the experiment in the laboratory. He took a good, healthy, normal chicken and injected this particular bacteria. And he found that yes, the bird developed fowl cholera and then died. So that part established Louis Pasteur's contribution and established that yes, this P. maltosida is actually responsible for fowl cholera. Isolating or finding out that which particular microorganism is responsible for a disease is very much important, because otherwise how you develop medicine if you do not know the causal organism. So that was a great job. But then after doing this job, a story goes on like this. You know, sometimes the historical stories are maybe only the stories, myth, but we like to hear those stories, you like to remember those stories in that way. And that's the way it has been described in the textbook also. So after this experiment is done, Louis Pasteur went for a summer vacation for 2 months' time or so. At that time, he asked his assistant to culture the bacteria. So maybe you do not have much idea about culturing bacteria. So in laboratory, we can culture bacteria. Culture means we can keep the bacteria alive in an artificial medium. We take a round type of plate, which is known as Petri plate, after a scientist Petri. And then we put some food material, which is known as Luria, Bartini agar, nutrient agar, etc. Because we already know that what type of food a bacteria loves very much. So we make a semi-solid jelly-like plate full with a jelly-like substance. You can imagine a plate with a full jelly-like substance. Maybe I will try to show some pictures sometime. And then we can take a bacteria, look full of bacteria in a small needle or something like that. This is called an inoculation needle. There is a ring of wire, small ring. So you can touch any surface of the bacteria and then spray it in the plate. So if you keep the plate at 37 degrees centigrade most of the time, that is our body temperature, the bacteria will grow and will remain there. You can see there's a growth of bacteria, but you cannot see the morphology. You cannot see the bacteria moving or anything. Just a line, you can see the bacteria there. Okay, so but you know, this media, as I said, nutrient agar, luria agar, the food, those food material contains a good amount of water. So if you keep the plate in the laboratory, in the room temperature, gradually the water will be evaporated. And within 2 months, 3 months time, all the water will be gone almost, and the media will be dried up. And most of the bacteria also will be almost in the verge of dying. They will also die because there is no water. So what do? We generally keep the plates into the refrigerator at 4 degrees centigrade temperature, which minimizes the chance of evaporation. Or what we do, we continuously culture the bacteria into new plate. Every 2-3 days after, we do a subculture from one plate to another, to always keep the fresh bacteria in the laboratory for our work. So exactly that Louis Pasteur asked his assistant to do. But somehow the assistant did not do that. He forgot or something like that. So here, after returning from a summer vacation, Louis Pasteur only found that some old dried culture of bacteria is there. No new fresh culture is there. Anyway, nothing can be done. Then either Louis Pasteur has to isolate the bacteria once again from the foul cholera infected chicken, from their excreta. But before doing that, he did a small experiment. He used that old culture to inoculate a fresh, healthy, normal chicken. So what is the difference? In the first place, it was a fresh Maltosida bacteria, and here, in this case, it's aged, old bacteria. Okay. In the first place, in the first place, we've seen that the chicken is developing foul cholera disease and dying. Here, to the surprise of Louis Pasteur, the chicken survived. They didn't have any disease. Only difference is that the bacteria were aged. They're not all died, but they're aged, old culture. So that is a surprise to Pasteur. But the reason still not clear. You know, there could be a lot of different type of explanation could be there. Well, most of the bacteria were dead, so it was kind of expected or something like that. Then another twist of this story goes on. So now Louis Pasteur once again isolated fresh culture, fresh Maltosida he now have. But what he didn't have is a fresh batch of chicken. He's running out of chicken. We do not have enough experimental animal in his laboratory. So now it's a problem. So now what he did, he used this chicken because this chicken is survived. So he can use this chicken for his next level of experiment. He exactly did that. So if you follow this picture, normal chicken inoculated with aged or old Maltosida bacteria, we know the chicken survived, no disease or death. The same chicken is now used to inoculate another round of fresh Maltosida bacteria. What we have seen before, we know that whenever a chicken is injected with a fresh bacteria, he or she must die. But here, to the surprise to the pastor, the chicken survived again. So what is going on with this chicken? Neither they're getting died with the aged culture, not they're getting they're dying due to the fresh culture. That was a very surprising observation, experimental result found by Louis Pasteur. As the story goes, his supply of chicken was limited and therefore used the previously injected chickens. Again, to his surprise, the chickens were completely protected from the disease. They are not dying. So only difference what is there, that these chickens were already inoculated with the aged one. That is the only difference between this chicken and another normal chicken. Fine. So this is a full flow chart, as I'm saying. So if you use the fresh Multisoda, it will die. But if you use the fresh Multisida blood, it is not dying when you're using a chicken which are already inoculated with the aged bacteria. Okay. So Pasteur now hypothesized. Hypothesis means based on some observation on some basic experimental result, you can make some hypothesis. Hypothesis, then you have to prove that experimentally again. But hypothesis is, of course, always our basis of thinking. By seeing something, you can hypothesize. By seeing something or observing, you can hypothesize again that no, Earth is not revolving against the sun, sun is revolving against the Earth. So that is only a hypothesis you can give, then you have to prove it. So Pasteur hypothesized that what is going on here? He said that whenever we're using the aged bacteria, Maltosida, aged bacteria, aged culture actually weakened the virulence. Virulence means the power to cause the disease. As the bacteria was surviving in the plate for a long time without water or nutrients, etc., they become very weak, durbal, and the virulence of the disease-causing power of the pathogen become less. So that is quite natural. So that's why this type of chicken, this group of chicken survived. Well, you understand that. And now he used a term called attenuated. Attenuated means weakened, not strong enough. But found that the bacterium here added is not dead. They're alive, but they're weakened, very weak. So that means those weak bacterium entered into this chicken's body, could not manifest the disease, but when fresh Multisoda has been given, they're giving a protection. So it's something like what Edward Jenner observed at the time. So people who are surviving with the cowpox is getting protected against smallpox, something like that. So if a chicken animal is surviving against P. Maltosida, then they are protected against the actual fresh culture or fresh Maltosida who has good amount of disease-causing power. They're getting protected. So such an attenuated strain, so that's why Pasteur thought that this observation or this result could be extended to the other disease as well. If we can make a bacterium or virus weak somehow, then we can use that bacterial virus, which we call attenuated strain, to protect the animals against the actual disease. Once again, if we can somehow weaken a particular virus or bacteria, or if we can limit the disease-causing power, in actual term, we can attenuate a strain, and then if we administer or inject that strain to an animal, then the animal could be protected against the actual bacteria, the fresh one, the natural one. So that was Pasteur hypothesis, and that was a remarkable and very important hypothesis. And Pasteur, this is the basis of our vaccine concept. Pasteur used the term vaccine for the attenuated strain. Actually, it has no meaning. The term vaccine is coming from a Latin word vacca, means cow. Why? Because Pasteur was very much respectful to physician Edward Jenner. And if you remember, Edward Jenner did work on cowpox. So just to give a homage, respect to the Jenner, Pasteur used the word vaccine. Okay, and that was the first observation or experimental proof that this way vaccine could be developed. Okay, so later Pasteur extend these findings to, as I said, to other works, other diseases like anthrax, etc. He found that how to attenuate or weaken a pathogen and then administer. Say, for example, I said anthrax. Anthrax is caused by a bacterium known as Bacillus anthracis. Anthrax generally do not infect people, do not come to the people. It only more or less restricted to the cattle. That's why the people who come into close contact with the cattle, such as cattle breeders, such as milkmaid, as I said, butchers, etc., they can get affected with the anthrax. That's a nasty disease. But at that time, the main concern was not about human. Concern was how to protect our cattle, because that was a source of economy. So that's why anthrax disease, people are very much requesting Dr. Pasteur to do something about the anthrax. So he thought his earlier knowledge could be used here for the anthrax disease. So what he did, he used heat to the anthrax bacillus organism. Not boiled actually, but he used 80 to 90 degree temperature. So anthrax bacillus, pure culture form of bacteria he had, he applied some heat to this anthrax bacillus and then injected that heat treated anthrax that we can call now attenuated heat treatment. We know from our very, very common sense or knowledge for a long years that heat can destroy the bacteria or different types of microorganisms. So that was also the concept here. So if you apply the heat, maybe the bacteria is not dying all the way, but they're getting very weakened. So those are attenuated. So now inject those to a group of sheep and consider that they are now vaccinated. Fine. Okay, now what he did, later he challenged the vaccinated sheep, challenged the word used in immunology when you inoculate or inject some antigen or some pathogen. So here you can see two groups of animals we have: vaccinated sheep, unvaccinated sheep. Something like you can see the corollary now we have COVID vaccinated human being, COVID unvaccinated human being. Okay. So vaccinated sheep and unvaccinated sheep. So now Pasteur injected the virulent culture, not the attenuated one, not the heat treated, the fresh virulent anthrax culture injected to both the group of sheep. Okay. So what was the expectation? Expectation is that the vaccinated one will survive and unvaccinated one will die. And exactly that happened. All the vaccinated sheep remained alive and all unvaccinated sheep died. So once again, Pasteur proved it experimentally that his observation or hypothesis is actually correct. So what was his hypothesis? Hypothesis was that if you can weaken the pathogen either by aging or by heating. Later we'll see if we can do that with some chemicals too. Then the attenuated strain could be administered to an animal to protect against the actual disease. You only have to imagine here that here is an artificial situation. You are infecting the animals with a virulent culture intentionally, but you have to replace that part, natural infection. So unvaccinated sheep and vaccinated sheep, both groups, if you are exposed to the natural infection, this group will die and this group will survive. So that was also a remarkable journey, a remarkable discovery in terms of the immunology. So some people believe that this experiment marked the beginning of the discipline of immunology. Jenner's work and then Louis Pasteur's first work about the fowl cholera, the accident, etc. And then when he extended the experiment to the sheep for the Bacillus anthracis or anthrax disease. Okay, so I would say that's all for the first day. I do not have much to say for the first day now. If you have any question at this point, you may ask. If you have any questions,
Sir.
Yes.
Sir, agriculture department UID 003 অনন্যা চক্রবর্তী স্যার ওনা অ্যাটেন্ডেন্সটা দিতে পারেনি।
অনন্যা চক্রবর্তী।
Yes sir.
Okay.
হ্যালো স্যার।
হ্যাঁ হ্যাঁ বলো কে বলছো?
অনন্যা চক্রবর্তী স্যার বারবার স্যার কলটা না ড্রপ হয়ে যাচ্ছে নেটওয়ার্কের জন্য। এর জন্য স্যার তখন লিংকও খুলছিল না।
Okay, I will provide it. No problem.
Okay sir, thank you sir.
আর কোনো প্রশ্ন? Attendance related প্রশ্ন ছাড়া? And once again, I would like to ask that this was the first day, so it would be better if you can give me your feedback. How is it going? Am I going too fast or something like that? So I will adjust accordingly.
Yes sir, like একটু ফাস্ট হয়ে যাচ্ছে for
একটু ফাস্ট হয়ে যাচ্ছে। আচ্ছা, কে বলছো বলো, আমি নামটা দেখতে পাচ্ছি না। সুপ্রিয়া, সুরাইয়া, কে বললে কথাটা? কমেন্টটা বলো।
সোফিয়া খাতুন।
সোফিয়া খাতুন বলো। আচ্ছা, ওকে, গুড, আই উইল কিপ দ্যাট ইন মাইন্ড। এনিথিং এলস? তো মোটামুটি বোঝা যাচ্ছে যা বলছে?
Yes sir.
Yes sir.
Okay.
Yes sir.
Good,
Yes.
very good. So হ্যাঁ বলো, কিছু বলছো? So initially there was 46 students, then after attendance some of them are left the class. That is more or less an usual practice. I have no control over that. I can see 35 now out of 54. So that is up to you. If you enjoy the class, you can come and join in the next week. If you do not enjoy, that is up to you. I'll provide the attendance, everything. Let's see how many students have provided attendance here in the list. 43, keeping on to be 44. Okay, that's okay for today. We'll be communicating through the WhatsApp group, and most probably see you in the next Wednesday at the same time. Okay.
Thank you sir.
So with that, I'm signing off for today.
thank you.
Okay. And
Thank you, sir.
Thank you.
স্যার আপনি কি লিংকটাকে আবার